Hello students, today we will be dealing with the topic electron transport chain. I am Shaista from Department of Studies in Microbiology KSOU. We will be dealing with electron transport chain in detail, studying its various components as well as complexes involved in this chain and its stepwise reactions as well as ATP synthase. Okay, so let's begin with this class. So students, how is the electron transport chain defined as? Electron transport chain is defined as a series of reactions that converts the energy which is released from the oxidation of NADH and FADH2 into proton motive force, which is in turn used to synthesize ATP through many conformational changes that is occurring in the ATP synthase complex. And this entire process is called as oxidative phosphorylation. Now, let's break the definition into single parts and understand what actually it means. So, to understand this, the electron transport chain is converting the energy which is which is available from the oxidation of NADH and FADH2. So, when NADH is oxidized to NAD and FADH2 is oxidized to FADH, there is a release of energy in the form of electrons. Okay, and these electrons are transferred from one complex to the another complex, and when the transport of this electron is occurring, there is a movement of hydrogen protons from inner matrix of the mitochondria to the outer matrix, and this is nothing but the proton motive force. Okay, the proton motive force which is used to synthesize the ATP. So when there is a higher concentration of hydrogen over here, the proton motive force increases, and then there is an inlet of this proton motive force which helps in the synthesis of the ATP. Okay, and this entire process is called as oxidative phosphorylation. And where are the sources of this NAD, FAD and everything is coming from? We are getting 3 NADH from the TCA cycle, 1 FADH from the TCA cycle, and pyruvate also results in NADH. Fatty acid synthesis, that is fatty acids breakdown, beta oxidation also results in acetyl CoA, and this in turn is used up in the TCA cycle for the generation of FADH2 and NADH. And thus these all are used in this electron transport chain to generate the ATP. I hope you are clear with the definition now. So students, now let's see the important points of this electron transport chain. The oxidative phosphorylation is common and last step in the cellular respiration. It is common for all the degradation of carbohydrates, fats, amino acids, where they are converted into energy, that is the synthesis of ATP, and this is the final oxidation step for all the molecular substances like carbohydrates, polysaccharides, fats and amino acids. So this cycle is this chain is very important. This change involves the uh, series of electron transport from organic compounds to the oxygen while simultaneously releasing energy during entire process. In case of aerobic respiration, the final electron acceptor is molecular oxygen, but in case of anaerobic respiration, the final acceptor is like sulfate. This chain involves large protein complexes, mainly 4 protein complexes called 1, 2, 3, and 4. They are embedded in the inner mitochondrial membrane. Together, they form the respiratory chain or the electron transport chain. This reaction helps in continuously generation of ATP. When the body utilizes the ATP, the electron transport chain is able to supply the ATP, which is in constant demand by the cell. So these are the important points of the electron transport chain, and this is the final oxidative pathway or oxidative degradation pathway for all the organic compounds within the cell. Now let's discuss about the location of this electron transport chain. So students, where is this chain located? Or where does this chain occur? This chain occurs in the mitochondria. Mitochondria is a double membrane organelle that consists of an outer membrane and an inner membrane, and that is folded into series of ridges called as cristae. There are two components in the mitochondria, that is the matrix and the inner membrane. The outer membrane is highly permeable to ions. It contains enzymes necessary for citric acid, while the inner membrane is impermeable to various ions. It is not permeable and contains uncharged molecules, electron transport change, and ATP synthesizing enzymes only. Okay, the number of electron transport chain in a mitochondria depends on the location of mitochondria and the function of the cell within which the mitochondria is existing. Suppose let's consider the example of liver mitochondria. There are nearly 1,000 sets of electron transport chain. Now, when compared to the heart mitochondria, it has 3 times the number of electron transport chain, that is 30,000 sets in the heart mitochondria when compared to the 1,000 sets in the liver mitochondria. The inner membrane space contains the enzymes like adenylyl kinase, and the matrix contains the ATP, adenosine triphosphate, adenosine monophosphate, NAD, NADP, and various ions which helps or acts as a cofactor in the electron transport chain. Now we are studying the various components involved in electron transport chain. These are not the complexes but the components. We will deal with the complexes later in ahead in this class. Now let's see what are the three major components of electron transport chain. They are called as FMN molecule, that is flavin, mononucleotide, ubiquinone or the coenzyme Q, and the cytochromes. Now let us deal with each one in detail. FNM, that is flavin mononucleotide complex or the components, they are found usually the beginning of the electron transport chain. They transfer the electron from NADH to FNM molecule, resulting in the formation of FMNH2. There is a transfer of the hydrogen from the NADH. Okay, when NADH is oxidized to NAD, there is a transfer of the hydrogen from the NADH complex to the flavin mononucleotide complex. Okay, this is catalyzed by the enzyme NADH dehydrogenase. Okay, and further, these electrons from FNM is carry forward to a series of iron sulfur complexes, that is even called as FES complexes. These complexes have greater affinity towards these electrons. Okay, now let's deal with the other component. So our next component is ubiquinone. It is also called as coenzyme 2. This is located between the flavoproteins, that is nothing but FNM component, and the cytochrome. It is located between these two. This is the only electron carrier in the respiratory chain that is not bounded or attached to any protein. Therefore, it moves freely between the flavoproteins and the cytochromes. Okay, now let's see the equation. When there is an electron transfer occurring from the flavoprotein to the ubiquinone, the ubiquinone is converted into ubiquinone hydrogen complex, and the FNMH2 is oxidized to form FNM, flavoprotein, which is released in this oxidized form so that it can again go ahead and carry forward the first reaction of the electron transport chain. Okay, I hope it's clear now. Students, the next component of electron transport chain is the cytochromes. Cytochromes are the electron carriers, which are generally red or brown color proteins. They contain a hem group which carries the electrons in sequence from ubiquinone to the molecular oxygen. However, you should know the fact that each cytochrome will carry a single electron, but whereas an FNM, that is flavoprotein or ubiquinone, can transfer two electrons. So this is the basic difference between a cytochrome. It has an ability to carry a single electron or to transfer a single electron only. There are five types of cytochrome which are located between ubiquinone and the molecular oxygen, and they are A, B, C, D, E. These are classified based on the ability to observe the wavelength, different wavelengths of light. Yeah, A having the least ability to observe the least wavelength, and whereas the E cytochrome has the highest ability to observe the longest wavelength. So students, now let us see at the diagrammatic representation of electron transport chain, and especially the location of these uh, complexes on the mitochondrial membrane. So in the figure you can see this is the cytoplasm, and this is the outer membrane of the mitochondria. This is the intermembrane space, the inner membrane, and the mitochondrial matrix. So where are the complexes located? The complexes are located on the inner membrane of the mitochondria. This is complex 1, complex 2, complex 3, complex 4, and the ATP synthase complex. This is ubiquinone and this is cytochrome C. So this entire thing together forms the electron transport chain. We will see how this flow of electrons is occurring through these complexes in detail as we proceed further with this class. Okay. Students, now let us study about the complex 1. It is mitochondrial complex 1. This the first complex over here. This is also called as NADH dehydrogenase complex. It contains FES centers and it even contains FNM, that is flavoproteins associated with this. So what does this enzyme complex do? This enzyme complex facilitates the oxidation of NADH into NAD plus H generation of H ions. Okay, this is a two-step process. It is happening in presence of a flavoprotein where NADH is transferring its hydrogen to FNMH2 complex, that is flavoprotein hydrogen complex, and then the electrons are transferred to the FES centers, and then it is transferred to the ubiquinone. Okay, the electrons are transported, not the hydrogen. The hydrogen gradient simultaneously, for every two transfer of the electrons to the ubiquinone, this hydrogenase complex transfers four protons, that is four hydrogen ions, into the intermembrane space. There is an outflow of four hydrogen ions into the intermembrane space where two electrons are transferred to the ubiquinone complex. Okay, this you have to understand, and these two are occurring simultaneously during the oxidation of NADH into NAD. Okay, this is happening at the complex 1 of the mitochondria, which is also called as NADH dehydrogenase complex. So students, now let us see the second complex. This second complex is also called as succinate dehydrogenase complex. It consists of succinate dehydrogenase and FES centers. These complex 1 and complex 2 run simultaneously and parallelly to each other. This captures the electrons when FADH2 is oxidized to the FAD. It captures the electrons and transfers to the ubiquinone. But there is no outflux of hydrogen into the intermediate intermembrane space as you see in the complex one. There is only the transfer of electrons to the ubiquinone, but no transfer of the hydrogen ions. Okay, this is the difference between the complex 1 and complex 2. Okay, this you can see over here. Only an electron transfer is occurring between the complex 2 and the ubiquinone. Students, now let us look at the third complex of the mitochondria, that is cytochrome reductase complex. It consists of various cytochrome like B, C, and A, and it even consists of FES center. When the ubiquinone receives the two electrons, this complex helps in the transfer of the electrons to the cytochrome C. And meanwhile, it also facilitates the outflux of four hydrogen ions into the intermembrane space, creating the proton gradient. Now, when NADH is getting oxidized to NAD, the ubiquinone is forming an ubiquinone hydrogen complex, which can be oxidized back to the ubiquinone complex component in presence of iron ions. So this is done in the cytochrome C complex in presence of ferric ions. And meanwhile, there is a resultant force of outflow of four hydrogen ions into the intermembrane space, creating the proton gradient. Now, students, let's deal with the fourth complex in the mitochondrial membrane, inner membrane. This fourth complex, it consists of cytochrome A and A3, which is also called as cytochrome oxidase. This is the last complex of this chain and is involved in the transfer of two electrons from the cytochrome C. These two electrons are transferred to the molecular oxygen, forming water. You can see there is a transfer of these two electrons to the molecular oxygen, and combining with the two hydrogen ions, it's forming water. In meanwhile, four protons are translocated across the membrane. So there is an outflux of four protons again into the intermembrane space, creating a proton gradient. Okay, so these are the four complexes where there is an electron flow from complex 1 to the ubiquinone 3, then to the cytochrome, then to the complex 4, and then to the molecular oxygen, where hydrogen ions combine with the molecular oxygen in presence of these electrons forms the water. Okay, now with all this transfer, there is each time transfer of four hydrogen ions from each complex, like 1, 3, and 4. Therefore, in intermembrane space, there is high concentration of proton gradient. Now, what will happen to these high concentration of proton gradient? We will see in the next slide. So in this diagram, this is an inner membrane and here are four complexes, and a fifth complex, I can say, or the fifth complex, which is the most important complex, ATP synthase, is located over here. This is where ATP is synthesized. Now for every time there is a two electron transport. With two electron transport, four hydrogen output is occurring. So for every single electron, there is an output of two hydrogen. Okay. When there is a single electron flow, there is an output of outflux of 2 hydrogen. When there are 2 electrons, there is outflux of 4 hydrogen. Okay, so for every time there is an outflux of 4 hydrogen occurring over here. Now, when there is a high proton gradient in the intermembrane space, what happens? This results in the proton move back into the matrix through a pore in ATP synthase. These protons, because of the high proton gradient created here, there is an influx of these proton ions through a pore in this ATP synthase into the matrix. And while this is happening, this results in the formation of energy, which results in the substrate level phosphorylation, converting the ADP adenosine diphosphate into the ATP adenosine triphosphate. This cycle is so efficient that whenever there is demand for the adenosine triphosphate by the cell, the ATC electron transport chain is meeting all the demands, as electron transport chain is the final oxidative step for all the organic metabolites. uh, the energy is generated over here in form of ATP, thus meeting the cell energy needs. So, because of the continuous generation of the ATP in the mitochondria, which meets all the cell energy needs, thus the mitochondria is called as the powerhouse of the cell. Now, I think you have understood. This is mainly because of the electron transport chain, which is common for all the oxidative cycles, which is the end pathway for all the oxidative cycles over here. Now let's look at the electron transport chain equation. The electron transport chain equation consists of a series of oxidation reduction reaction that leads to the release of energy. Thus, this can be summarized as NADH in presence of molecular oxygen and hydrogen, and adenosine triphosphate and phosphate results in the oxidation of NAD, oxidized form of NAD, the energy rich form of ATP, that is adenosine triphosphate, where this combination has occurred here, and those two electrons are accepted by this molecular oxygen in resulting forming of water molecule. Thus, the energy generation and formation of the water molecule. I hope now you are clear with the electron transport chain and the earlier definition of electron transport chain, which I have told you. I have explained it in detail by using various complexes and how the electron flow is occurring and how the water formation is happening and how the ATP is synthesized. I hope you have understood. Okay. So thank you, students, for patiently listening to my class. I hope I made it easy for you to understand the electron transport chain. I'll be dealing with other cycles ahead. Thank you so much.